Hi everyone, my name is Pia and I'm a second year PhD candidate at Monash University in Australia. A big thanks to the conference organisers for giving me this opportunity to present my research on whole blood methylation as a prognostic biomarker in multiple sclerosis. So what is MS? It's a chronic inflammatory and neurodegenerative disease, and it's characterized by pro-inflammatory peripheral immune cells translocating into the central nervous system and causing demyelination. This neuroinflammation and demyelination usually occurs in the white matter of the central nervous system and shows on MRIs, as you can see here, as a brain lesion. Depending on the location of these lesions, MS patients experience a wide array of symptoms. MS is important because it affects over 2.8 million people globally, and it's currently the most common cause of neurological disability in young people. It also mainly affects women in their reproductive years, as 75% of patients are women diagnosed between 20 to 40 years of age. In MS, disability is measured using the Expanded Disability Status Scale, or EDSS score, and this is a non-linear scale from 0 to 10, which is routinely used in the clinic and in clinical trials and recorded in MS-based registry, which I will touch on later. And so with the EDSS score, a score of 0 refers to normal neurological function, and 10 is death due to MS. Related to this is the age-related multiple sclerosis severity, or ARM score, and it's the EDSS score normalized for age, and it's actually been shown to be more consistent over long periods of time. And so this is what we used in this study to characterize disease severity. So how is MS managed? Currently, the biggest challenge in MS care is that disease activity and progression are highly variable between patients. And this makes it very difficult for doctors to pick the right treatment for the right patient at diagnosis. And so we know that effective treatment immediately after diagnosis gives patients the best long-term outcomes in terms of disability. But if doctors don't know how severe a patient's disease is going to be in the long term, they increase their risk of disease progression if they put them on a therapy that's too mild, or alternatively, if they put them on a therapy that's too strong for their mild disease, they unnecessarily expose them to quite severe side effects. And so there is a critically unmet need in MS care for prognostic biomarkers that inform treatment management. So we hypothesized that DNA methylation patterns will differ between patients with mild and severe MS, and this can be harnessed as a prognostic biomarker. So how did we get to this? Well, even though there are over 200 genetic susceptibility variants for MS, currently 0 variants associated with disease severity have been validated. And even though there are some published studies, in these studies, genetics explains at most up to 33% of the variation in severity in the studied cohorts. And so this tells us that MS disease severity is most likely regulated by an interaction of genetic and environmental factors. Now, epigenetic mechanisms regulate gene expression and are impacted by environmental factors. And currently, to the best of our knowledge to date, no epigenetic predictors of disease severity have been identified in MS. DNA methylation is one type of epigenetic mechanism, and it regulates genes. It's a genome modification whereby a CH3 or a methyl group is added to a CG dinucleotide on the DNA sequence. And it regulates gene transcription in a manner where methylation in gene promoter regions suppresses gene transcription. It's also cell and tissue type specific because it affects phenotypes, and it's also highly dynamic and reversible. So it's impacted by developmental stage, environmental factors, and is also an attractive therapeutic target as it's reversible. So for this study, we wanted to know, 1, if whole blood methylation is associated with disease severity, 2, if whole blood methylation can be used as a predictive biomarker of disease severity, and 3, if immune cell subset DNA methylation is associated with disease severity. So for this study, we use clinical and demographic data from MS-based registry. And it's a prospective and longitudinal patient outcomes database that contains records for over 75,000 neuroimmunological patients worldwide. And the majority of these records are for patients with MS. So from MS space, we identified patients with relapse onset MS, and we categorized their disease severity using the age-related severity score. And those that fell in the bottom 20th, below the 20th percentile were categorized as having mild disease, while those above the 80th percentile were categorized as having severe disease. And these are the patients that we targeted for recruitment. So we successfully recruited over 1,000 of these patients, and then we genotyped and biobanked their DNA. And then for this specific study, we used only Australian females who also had genetic data available for MQTL analysis. And so this left us with 235 patients, 119 of which had mild disease, and then were age matched with 116 who had severe disease. So DNA was extracted from these patients using whole blood samples, and it was bisulfite converted before measuring methylation using the Illumina EPIC array. And this array measures methylation at over 850,000 CpG sites. And it gives us a beta value, which is an estimate of the methylation at this CpG site, and it ranges from 0 to 1. So for methylation data pre-processing, we used the chip analysis methylation pipeline, or CHAMP in R. And so we used beta values, did standard probe filtering, and this left us with over 747,000 probes to analyze. We normalized our data using champ.norm, and we used the beta mixture quantile method. We then did batch effect identification and correction using singular value decomposition and also combat. And you can see that we removed the majority of our batch effects. So this then gave us an analysis-ready beta value matrix. Before I move on to our key results, I will just flag some key definitions. So differentially methylated positions, or DMPs, are a single CpG site with different levels of methylation between mild and severe groups. A differentially methylated region is a genomic region with multiple DMPs with the same direction of effect. So all of these DMPs have a higher level of methylation in the red group than in the blue group. So moving on to differential methylation analysis, we did this again in CHAMP using linear models to identify DMPs. And this is subject to multiple testing burden. So we used an FDR of 0.05 as our threshold. We also prioritized DMPs with an effect size above 1%. We then performed GSEA analysis using TopGene and GoMeth as part of the MIS-methyl package, and then conducted sensitivity analyses to investigate the effects of covariates on methylation. We found no association between our covariates, and so we didn't adjust our linear models to identify DMPs for any variables. We also conducted DMR analysis using two methods. The first was DMR-CATE within CHAMP, and the second was an alternate paradigm where we took our list of DMPs and identified DMPs that had an FDR below 0.01, the same direction of effect, and were located less than 1,000 base pairs apart. And we defined a DMR as a genomic region that had at least two of these DMPs meeting that criteria. So overall, we found that whole blood methylation is associated with disease severity. We identified over 500 DMPs, most of which had small effect sizes, which suggest a cumulative effect on disease severity. Most of our DMPs were hypomethylated in the severe group, shown here in blue. Although I guess it was roughly half-half, some were hypermethylated in the severe group. Conducting gene set enrichment analysis, we found that differentially methylated genes were enriched in central nervous system related cellular components and pathways. And this is consistent with our current knowledge of severe or progressive MS, where we know that it's driven by neurodegeneration as opposed to autoimmunity and neuroinflammation. And so this is consistent with what we know. In terms of DMRs, we didn't identify any DMRs using the DMR-CATE algorithm, but using our next paradigm, we found two DMRs, each containing two DMPs. and so one located in chromosome 11 and one in chromosome 15. Unfortunately, these DMRs were located either or mapped to an uncharacterized long non-coding RNA or intergenic region, and so we didn't gain much mechanistic insight from these DMRs. Moving on to aim number 2, to determine if whole blood methylation can be used as a predictive biomarker. So to do this, we use machine learning to train an elastic net regression model. And so we trained the model on 70% of our cohort and then tested it on 30% of our cohort to reduce overfitting. And so the input data was the methylation data, so our beta value matrix. And our final model contained 890 CpG sites, and 350 of these overlapped with our previously identified DMPs. These genes were also enriched in nerve growth factor signaling pathways, which again highlights the role of central nervous system function in disease severity in MS. So we tested our final regression model on the remainder of our cohort, and we got an area under the curve of 0.91. So what this means is that using methylation levels at 890 CpG sites, the model could accurately predict if a patient had mild or severe disease with an accuracy of 91%. Moving on to aim number 3, immune cell subset DNA methylation. So we predicted cell type proportions using EpiDish, which is a reference-based method. And we then performed statistical deconvolution with cell DMC to find cell-specific DMPs. We found no differences in cell type proportions between our mild and severe groups, and also no cell-specific DMPs using statistical deconvolution. So what did we find? Overall, we found that whole blood DNA methylation is associated with severity in a manner where small effects are probably cumulative. We found that whole blood methylation can accurately predict disease severity, and that cell-specific methylation isn't associated with severity, and so methylation is likely to be systemic. The limitations of our study include that we only studied Australian females, and so this limits generalizability. And also statistical deconvolution is not as accurate as performing physical cell isolation to identify cell-specific differential methylation. Overall, I would like to thank everyone involved in the study, supervisors, our lab group, and of course the patients and our funding bodies, particularly Multiple Sclerosis Research Australia. Thank you.